This set of slides talks about the logical properties of connectrons. Connectrons, remember, act where the source is a source of transcription, whether it be gene coding or non-coding, and then there's a target region that forms a loop. So here's one source forming a loop here, but then on the same chromosome there can be another or many other sources that also form the same connectron. So this is called the many-to-one property. Of course, you can also have one source radiating control to many different places, and that's the one-to-many property. But it doesn't make any difference whether the sources are on the same or different chromosomes than the targets. And so these are just logical properties of connectrons, or the connectron idea doesn't matter to the connectrons where the sources and targets are in terms of chromosomes. They're just DNA. So this is the logical ending. So here's a transcription source, and it has the effect of forming a loop here. But this element, or an element here, let's say, was also generating a connectron here. So when this thing transcribes, it has the effect of turning this off. But when this thing is turned off, then it no longer makes this. And so the logical effect of this transcription is to turn on this element here. So instead of just being a negative operator, a pair of ANDed connectrons can be a positive operator. Of course, you want to be very careful as you do that. Okay. Of course, it doesn't matter which element is on which chromosome, since it's all just DNA. So the intra-chromosomal connectrons are the ones where the source and target are on the same chromosome. The greatest number of connectrons are formed by sources that are on the same chromosome as the targets. The greatest number of connectrons in general are in mouse transcriptome, the X and Y chromosomes, which is interesting. So here is the diagram of the intra-chromosomal connectrons. And you see, in fact, chromosome 4 in mouse transcriptome produces a lot of connectrons, and the others here a little bit less, but X and Y produce an awful lot. But the background here, the 19 by 19 or 21 by 21, is not zero. It's just very low. So if we change the scale, then the diagonal elements, the intra-chromosomal connectrons, become huge telephone poles. And if we just logically eliminate these, you can see that this is the distribution that you get then. So this distribution tapers off in the direction of the length of the chromosome, as you'd expect. But in this direction here, it's random, it's fractal. And that's some indication that these connectrons are really real and really doing something to the behavior of the cell. And of course, each chromosome sends connectrons to X and Y. So if you knock out, logically, the 19 by 19 matrix of interchromosomal connectrons, then you see that each chromosome sends connectrons to X, which is blue, and to Y, which is pink, and that Y sends to X, and that it's only X that sends connectrons back to Y. Sorry, I'm just playing with PowerPoint there. Okay. So now, if we look at just the gene coding portion of this distribution, we see that it's low. It's a small portion, some smaller portion. Again, it's tapers in this direction, but random in this direction. And again, if we clip away logically the 19 by 19, then we see that as far as gene coding connectrons are concerned, that is, the gene coding means the source of the RNA is normally from an intron of a gene. that's the way what it really means, is that each gene coding element each chromosome sends gene coding connectrons to X and Y, and that Y sends to X, and X sends to the 19. Now, the same thing is true for non-coding elements. Non-coding elements are what were formerly called the pseudogenes, and now you see again the length dependent distribution, but random in this thing. In fact, you see a big, huge spike here. That is indication that that's something different and special. Again, if you clip out the 19 by 19, you see that each chromosome sends non-coding connectrons to X and Y. Y sends to X, and it's only X that sends back to the non-coding elements of the 19. So the most interesting observations are just what I've said. 1 to 19 send to X and Y sends to X sends to the 19, and Y sends only one chromosome to connectron to chromosome 7. So the one-shot connectrons are another logical property of connectrons. A one-shot connectron is a transcription element that turns itself off at some period of time. So it's just a burst of transcription and then a quiescent period. One-shots, by definition, are intra because the source and the target are on the same chromosome. One-shot connectrons can send additional connectrons to other transcription sites on the same or different chromosomes. And so it's clear from the statistics that the one-shots radiate control from themselves to many other genes and non-coding elements within the genome. So Sue Gardner from New Zealand, who I met at the Plant and Animal Genome meeting, suggested that I call these one-shots temporary self-knockouts. She said that biologists might understand this name more easily. Of course, since I trained as an electrical engineer, one-shots are parts of logical circuits, and so I tend to think of these one-shot connectrons in that fashion. So a one-shot, as I said before, is a promoter, some transcription thing that may or may not have introns in it, and if it has a signal C1 and C2, then that RNA acts on the flanks to generate a loop, and by forming two Hookstein helices that have a finite lifetime. The lifetime depends on the shorter of these two helices, because the short helix is going to boil away first, allowing the connectron to open up again. So the effect of a one-shot is you get a pulse of expression and then a delay as the loop is formed, and then the loop opens up again and you get a pulse of expression and then a delay again. So one-shots typically radiate control to other places on the same chromosome. These would be intra-radiations, and of course the one-shots could also radiate control to transcription sites on other chromosomes, and these things happen. Okay, and it's not just a question of that they illogically could happen. There are statistics to support all of these assertions. So here are the one-shot connectrons, the gene coding one-shot connectrons, and what you see is that the X chromosome has some one-shots on them, but the Y doesn't. And that if we go to the non-coding, well, here chromosome 4 has a huge spike of non-coding one-shot connectrons, and that X and Y have just a few non-coding elements. Well, not non-coding one-shots, I should say. So this is the complete logical diagram. That is, these are the gene coding transcription elements, and they can affect other gene coding transcription elements. And the same with the non-codings. They can affect themselves, other non-coding elements, different chromosomes, same chromosome. And of course they can send, so a non-coding can send a connectron to control a gene coding or vice versa. And then you have these one-shots, the things that turn themselves off, and they can in fact affect other one-shots. They can affect of the gene coding variety or non-coding variety, and vice versa. So you have this whole matrix of possibilities of interaction of different types of connectrons with each other, and there are statistics for each of these pathways, and these pathways occur in all genomes. When you have bacteria that have plasmids, well, then you have interactions between the chromosomes of the genome and the plasmids of the genome, also of the cell. So these logical pathways are the most important aspect, and they do occur. That's very clear. So one of the other things, the logical properties, is the idea of a transchromosomal connectron. So until recently, I I prohibited my program from finding these things because I didn't know what to do with them, and now I'm beginning to understand possibly what they could do. Transchromosomal connectrons finds a lot of these things, and it was because we were finding so many of them that I sort of cut them out arbitrarily. It turns out I shouldn't have done that. So a transchromosomal connectron is where there's a transcription source on one chromosome, and one of the targets may be on the same chromosome, but another target should be on a different chromosome. Or the source could be on one chromosome and the target on another, and the second target on yet another chromosome. So the effect of what should these transchromosomal connectrons do? Well, the ordinary intra or interchromosomal connectrons bring together pieces of one chromosome, whereas the transchromosomals would pull together pieces of different chromosomes in the nuclear space. So the Broad Institute has a paper on the structure of the human nucleus, and they show very nicely how many parts of the same and different chromosomes are in intimate 3D contact with each other. And so the transchromosomal connectrons might be a way, along with the condensin proteins, might be a way of helping to define the structure of the nucleus of a genome in 3 space. And that's what, of course, the transchromosomal connectrons are really no different than any others, because it's just some transcription source forming 2 triple-strand Hookstein helices that have the effect of bringing those pieces of DNA close together and keeping them there so they can be stabilized and even pasted shut. Thank you.